Let's talk about fixation artifacts. So fixation artifacts or pigments, these are unwanted deposits, discolorations, or changes in tissue morphology that occur during the fixation process. So these pigments form as a result of either chemical reactions between the fixative tissue components like proteins or blood and processing reagents, or due to improper fixation practices. Classical examples, or the commonest ones, we have formalin pigments, pink disease artifacts, chrome pigments, and mercury chloride pigments. Key characteristics of these pigments: one, they are typically seen as granular, crystalline, or discoloration deposits in tissue sections. Number two, they may interfere with staining and microscopic interpretation. And number three, their formation depends on the type of fixative and tissue handling. So let's go straight away to formalin pigment, also known as formaldehyde heme pigments or acid formaldehyde hematin. So formalin pigments are dark brown to black pigments that can form in tissues fixed in formalin. And these pigments are formed under the following conditions. One, acidic pH. So formalin becomes acidic over time due to the formation of formic acid, especially when stored poorly or not buffered. Number two, interaction with hemoglobin. So under acidic conditions, formaldehyde reacts with hemoglobin, leading to the production of formalin pigments. And this particularly happens commonly in blood-rich tissues or areas of hemorrhage. Number three, chemical reaction. For us to appreciate this pigment, it is as a result of the reaction between hemoglobin, formic acid, which is formed from acidic formalin, and formaldehyde. So classical features of formalin pigments: one, they are brown to black granular deposits in terms of appearance. Location, they are seen in areas where blood is present in tissue. And microscopically, we're going to see these deposits being birefringent under polarized light microscope. In this case, we mean that the pigment has the ability to refract light into two separate rays when viewed under a polarized microscope. Lastly, the pigments are usually extracellular but can also appear intracellular. So how do we prevent formation of these pigments? One, use of buffered formalin. Ideally, we use 10% neutral buffered formalin at pH 7.0, the fixative to prevent acidification. So what does buffered formalin do to prevent this pigment from being formed? One, when you look at formalin at 7.0 to 7.2, that's neutral pH. So buffered formalin contains phosphate buffers like sodium phosphate, monobasic, and dibasic. So these buffers stabilize the pH around neutrality. At this neutral pH, hemoglobin does not react with formaldehyde to form acid hematin, hence preventing the pigment from being formed. Number two, it minimizes acidic byproducts. So during fixation, formaldehyde can oxidize to formic acid, which lowers the pH of unbuffered formalin over time. So the buffered formalin neutralizes this acid, preventing the environment from becoming too acidic. Number three, optimal tissue fixation. Buffered formalin ensures better preservation of tissue morphology and staining quality. as it avoids the artifacts caused by formalin pigment. Point number 2, how to prevent formation of formalin pigments is proper fixation techniques or proper fixation. So avoid prolonged fixation and ensure the formalin solution is fresh and buffered. Why is it important to avoid prolonged fixation? 1. Accumulation of acid byproducts. So during fixation, formaldehyde oxidizes over time to form formic acid as a byproduct. So if fixation is prolonged, the amount of formic acid increases, making the formalin solution more acidic. Number 2, when the pH of the solution drops below 6.0, hemoglobin in red blood cells definitely react with the acidic environment to form the formalin pigment. In case these pigments have formed, how best can they be removed? 1. We can use saturated alcoholic picric acid, which usually falls under what we call Barrett's method. So saturated alcoholic picric acid, this solution has alcohol and picric acid. So what does alcohol and picric acid do in terms of removing the formalin pigment? 1. Solubilization by alcohol. So alcohol serves as a solvent to dissolve and extract organic components of the pigment from the tissue. Number 2, interaction with picric acid. So the picric acid acts as a weak organic acid, breaking down the chemical bonds in the formalin pigment. So it reacts with the ferric ions in the pigment, forming soluble complexes that can be washed away. Number 3, chelation of iron. Picric acid chelates the ferric ions from the acid formaldehyde hematine, which is our pigment, making the pigment soluble and facilitating its removal. So the second technique is alkaline alcohol. So in this case, we have two environments, alkaline environment and alcohol environment. So under alkaline environment, we have alkaline hydrolysis. So we have an alkaline environment. So the alkaline component, for example, sodium hydroxide or potassium hydroxide, breaks down the formalin pigment by hydrolyzing the bonds that stabilize the pigment structure. This leads to solubilization of the acid formaldehyde hematine. Number 2, alcohol environment. So in this case, alcohol serves as a solvent, which enhances tissue penetration and help dissolve the hydrophobic compounds of the pigment. Number 3, ion chelation and neutralization. So in an alkaline environment, the ferric ions in the formalin pigment are neutralized or precipitated, leading to pigment removal. Fact number 2, mercury chloride pigment. So mercury chloride pigment are dark to brown artifacts that arise when tissues are fixed using mercury-based fixatives. For example, Zenker's fluid or Haley's fluid. So these fixatives contain mercury chloride as a primary component. So mercury pigments are as a result of precipitation of mercury salts during fixation. So the mercury chloride in the fixative reacts with tissue components, majorly proteins, during fixation. How can these pigments be avoided, or how can we prevent them? 1, of course, avoid mercury-based fixative unless otherwise. In other words, we can use alternative fixatives such as neutral buffered formalin or other modern substitutes. Number 2, wash tissues post-fixation. After using mercury-based fixatives, it is a good practice to wash tissues thoroughly after fixation to minimize residual mercury salt. In case these pigments have been formed, 1. We can treat tissues with iodine. So iodine oxidizes mercury salts, turning them into a soluble compound. Number 2, clearing with sodium thiosulfate. So after iodine treatment, tissues can be immersed or sections in 5% sodium thiosulfate solution. This clears the iodine and removes mercury pigment. Number 3, we rinse in water. So wash tissues thoroughly with water after pigment removal.